Welcome, deep divers. Get ready for quite an exciting journey inside your own body today.
Yeah.
Because we're taking a deep dive into something incredibly fundamental, how your cells actually make energy. We're talking about glycolysis, your body's universal fuel pathway. Think of it as like the ultimate shortcut to power.
That's a good way to think about it.
We're going to explore how common sugars like glucose, fructose, and galactose are transformed into that life-sustaining energy currency, ATP.
That's right.
You'll discover why this pathway is so critical, and we'll even feature some fascinating clinical insights that show just how much these reactions impact your everyday health.
Absolutely. Our deep dive today is really inspired by a foundational chapter from Marks Basic Medical Biochemistry.
Okay.
And our mission really is to take these complex biochemical pathways and, well, distill them into clear, digestible insights.
It makes sense.
Exactly. Helping you feel truly well-informed without having to, pore over dense textbooks yourself. We'll guide you step by step through the key concepts, verbally illustrating these chemical changes and showing you the roles of the important players.
And connecting it to the real world.
All while connecting it to real-world examples that hopefully bring the science to life.
So let's set the stage. I've always heard glucose is super important for energy.
Yeah.
Why is it considered the universal fuel for pretty much every cell?
It absolutely is. Glucose is, without a doubt, the universal fuel. Almost every single cell in your body can generate ATP from it through this pathway, glycolysis.
Right.
It's important for, well, maybe two main reasons. First, glucose is just readily available. It's in our diet. It's always circulating in our blood.
Always there.
And second, glycolysis has this remarkable ability, this flexibility, to generate ATP both with and without oxygen. Your brain, for instance, relies almost exclusively on glucose. It's incredibly demanding.
Wow. Okay. And where exactly does all this magic happen inside the cell? Is it in the mitochondria?
Good question. Not the mitochondria, actually. All of the enzymatic reactions of glycolysis happen exclusively in the cytosol.
The cytosol?
Yeah, that's the fluid part of your cell, sort of outside the main organelles. Think of it like a small, highly efficient assembly line right there in the cell's main compartment.
Okay.
The overall process is quite elegant. One molecule of glucose, which is a 6-carbon sugar, right?
It's carbons.
It gets systematically broken down into 2 molecules of a 3-carbon compound called pyruvate.
Okay, so 6 carbons become 2 times 3 carbons.
Exactly. And this whole 10-step pathway ultimately generates a net of 2 ATP molecules directly. We call this substrate-level phosphorylation. It's like directly transferring energy from one molecule to ADP to make ATP.
Direct transfer. No messing around.
Right. And it also produces 2 molecules of NADH, which are crucial little shuttle molecules carrying energy in the form of electrons.
Okay, let's unpack this journey then. You said 10 steps. That sounds complicated.
It can seem that way, but it's actually quite logical. Glycolysis isn't just one big reaction, it's carefully orchestrated.
Right.
To make sense of it, you can really think of it in 2 main acts or phases. There's an initial preparative phase where the cell invests a little energy.
Like priming a pump.
Exactly like priming a pump, getting things ready. And then there's the ATP generating phase, which is where we get our big energy payoff.
Makes sense. Invest a little, gain a lot later.
Precisely. So in this initial preparatory phase, the cell does essentially prime the pump. First, the glucose molecule gets trapped inside the cell.
Trapped. How?
By adding a phosphate group to it. This step uses an enzyme called hexokinase. And it makes it so the glucose can't easily leave the cell. It sort of commits it to metabolism.
Oh, okay. Sticks a label on it.
Pretty much. And this new molecule, glucose 6-phosphate, is also a crucial branch point. It can go down glycolysis, or it can be used for other things, like making glycogen for storage.
Interesting.
And interestingly, your liver and pancreas have a special version of this enzyme called glucokinase, which has slightly different properties suited for their job in handling blood sugar.
So this initial phosphorylation is like ensuring the glucose stays in the game, ready for what's next.
Exactly. Then, in a couple more steps, isomerization and another phosphorylation, the glucose 6-phosphate is rearranged and another ATP molecule is invested.
Another one. So 2 ATPs used now.
Yep, 2 ATPs invested so far. This adds a second phosphate group, creating fructose 146 bisphosphate. And this second phosphorylation step, catalyzed by an enzyme called phosphofructokinase 1 or PFK1.
PFK1. Sounds important.
It is. It's often considered the first committed step of glycolysis. Once you make this molecule, you're definitely going down the glycolytic road. It's a major control point, highly regulated.
A point of no return.
Essentially, yes. Ensures the cell doesn't waste energy. Finally, this modified 6-carbon sugar is cleaved, split right in two by an enzyme called aldolase.
Into what?
Into two 3-carbon molecules. Dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate.
Okay, DHAP and glyceraldehyde 3-phosphate.
Right. And the DHAP is quickly converted into another molecule of glyceraldehyde 3-phosphate. So now we have 2 identical molecules ready for the next phase.
So we've invested 2 ATPs, cleaved the sugar, and now for every glucose that started, we have 2 glyceraldehyde 3-phosphates ready for the ATP generating phase.
This is where the cell starts to cash in, right?
Indeed, this is the energy payoff phase. Each of those 3 carbon molecules now undergoes a series of transformations.
Right.
First, it gets oxidized, loses electrons, and at the same time, an inorganic phosphate, just floating around in the cytosol, gets added.
Okay.
This step generates one of those NADH molecules we mentioned, carrying energy. And what's truly fascinating is that the enzyme involved here, glyceraldehyde 3-phosphate dehydrogenase, creates a really high-energy phosphate bond during this process.
High energy, like ready to make ATP.
Yeah.
Exactly. It allows for direct ATP synthesis, even without needing oxygen. This high-energy phosphate is then immediately transferred to an ADT molecule, generating our first ATP molecule in this payoff phase. Remember, this happens twice per glucose.
So 2 ATPs made right there. Nice.
Yep. Then, through a few more rearrangements moving the phosphate group around, removing a water molecule, another incredibly high-energy phosphate bond is created on a molecule called phosphenyl pyruvate, or PP.
EP.
This phosphate on PP is then also transferred directly to ADP by an enzyme called pyruvate kinase. This generates a second ATP molecule, and again, happens twice per glucose.
So that's another 2 ATPs, making 4 total in this phase.
Correct. 4 ATP produced in the payoff phase. Now remember, we invested 2 ATP back in the preparatory phase?
Right, 2 invested, 4 produced.
So the net result for one glucose molecule going through glycolysis is 2 pyruvate molecules, 2 NADH molecules, and a net gain of 2 ATP molecules.
Okay, net 2 ATP. That's the bottom line for just glycolysis itself.
That's the bottom line for glycolysis alone, yes.
So we've made pyruvate and NADH. What happens next? You mentioned it depends on oxygen. This sounds like a critical fork in the road.
It absolutely is. This is where glycolysis shows its versatility. If oxygen is plentiful and the cell's mitochondria are working well.
The powerhouses of the cell.
Exactly. Then the NADH's energy, those electrons it's carrying, get efficiently shuttled into the mitochondria. There, they power the electron transport chain, which ultimately leads to a much greater ATP yield.
How much greater?
We're talking around 30 to 32 molecules of ATP per glucose molecule when it's fully oxidized aerobically. The pyruvate also enters the mitochondria, gets converted to acetyl-CoA, and feeds into the TCA cycle, getting completely broken down to CO2. It's the grand prize for energy production. Super efficient.
30 to 32 versus just 2 net from glycolysis alone.
Yeah.
Huge difference.
Massive difference. But what if oxygen isn't so plentiful? Or what if the cell just can't use oxygen efficiently? Well, some cells naturally have limited oxygen supply, like parts of your kidney, or they have very few or no mitochondria at all, like your red blood cells. They can't do aerobic respiration.
Red blood cells have no mitochondria.
None at all. They need all their internal space for hemoglobin to carry oxygen. And of course, think about your skeletal muscles during intense exercise, like a sprint. The ATP demand just skyrockets, often outstripping the oxygen supply.
Right, you feel the burn.
Exactly. In these scenarios, anaerobic glycolysis kicks in. Anaerobic meaning without oxygen.
So what happens differently?
Here, an enzyme called lactate dehydrogenase, or LDH, steps up. It takes the NADH we generated earlier and uses its electrons to reduce pyruvate, turning it into lactate.
Pyruvate becomes lactate. Why?
This step is absolutely crucial because it regenerates NAD plus seri. Remember, NADH carried the electrons. LDH gives those electrons to pyruvate, freeing up NAD plus again. And NAD+ is essential for that earlier step catalyzed by glyceraldehyde 3-phosphate dehydrogenase to keep running. Without regenerating NAD+ seri, glycolysis would grind to a halt very quickly.
So it's a way to keep the glycolysis ATP production going, even without oxygen to take the electrons away later.
Precisely. It allows the cell to keep making some ATP, those net 2 ATPs per glucose, even in low oxygen conditions. The trade-off, though, is that much lower energy yield, just 2 ATP instead of 30 plus. And you end up with lactate as the end product instead of pyruvate going to the mitochondria.
And here's where we see the clinical impact, right? When this anaerobic pathway takes over, lactate or lactic acid, as people often call it, builds up.
Yes, and that can have significant consequences.
Right.
A classic example is someone like Linda F., a 68-year-old patient described in the text. She had acute hemorrhage and COPD, meaning she had very low oxygen levels in her blood, hypoxemia.
So her tissues weren't getting enough oxygen.
Exactly. They were starved of oxygen and had to rely heavily on that less efficient anaerobic glycolysis just to survive. This caused a massive surge in lactic acid production, leading to a dangerous drop in her blood pH, a condition called lactic acidemia, or lactic acidosis if it's severe. This buildup of acid explains her confusion and rapid labored breathing. Her body was desperately trying to blow off CO2 to compensate for the acidity.
Wow, that's serious.
It can be very serious. Or think about something much more common, like Ivan A's dental caries cavities.
Cavities? How does glycolysis cause cavities?
Well, certain oral bacteria, like lactobacilli and streptococcus mutans, absolutely thrive on dietary sugars, especially sucrose from things like candy or sugary drinks.
They love sugar.
They do. And they ferment these sugars using anaerobic glycolysis, converting them directly into lactic acid right there in your mouth.
Oh.
Yeah. This acidic environment then starts to demineralize your tooth enamel, literally dissolving the minerals away, leading to cavities. And some bacteria, like S mutants, even produce sticky substances called dextrans that help them cling to the tooth surface, keeping that acid concentrated right where it does the most damage.
So glycolysis in bacteria is bad for our teeth. It's fascinating how some of our own tissues are just built to depend on this anaerobic pathway, though. You mentioned red blood cells.
Right, red blood cells rely entirely on anaerobic glycolysis. No mitochondria means no aerobic option. Plus, they need to avoid oxygen interfering with hemoglobin's job.
And the lens of the eye.
The lens also has very few mitochondria. It needs to stay crystal clear to transmit light, and lots of mitochondria would scatter light, so it relies mostly on anaerobic glycolysis for its relatively modest ATP needs.
And then muscles during intense work, like Otto S doing wind sprints in the case study.
Exactly like Otto S. During those intense bursts, his muscles' ATP demand just goes through the roof, far exceeding what his aerobic system can supply immediately. Glycolysis ramps way up,
Generating ATP quickly.
very quickly, but also producing lots of pyruvate. Since oxygen delivery can't keep up, much of that excess pyruvate gets shunted to lactate by LDH. That lactate accumulation contributes to muscle fatigue and that burning sensation.
Burn.
And this also highlights the body's clever adaptations. In conditions where tissues chronically experience low oxygen, like Linda F. COPD or even high altitude,
Yeah.
a special protein, a transcription factor called hypoxia-inducible factor 1, or HIF1, becomes active. Think of HIF1 as a metabolic alarm system for low oxygen.
What does it do?
It turns on genes that increase the production of many glycolytic enzymes, boosting the capacity for anaerobic glycolysis. It also promotes the growth of new blood vessels to improve oxygen delivery. It's a crucial adaptation to help cells survive hypoxic stress.
That's incredibly smart. So what happens to all that lactate then? Does it just sit there making muscles sore or build up dangerously like in lactic acidosis?
No, the body is much smarter than that. It gets recycled. That's where another brilliant cycle comes into play, the Cori cycle.
The Cori cycle, okay.
Lactate released from cells doing anaerobic glycolysis, like muscles during intense exercise or red blood cells, travels through the bloodstream primarily to the liver.
To the liver.
Yes, the liver has the necessary enzymes to take up that lactate and convert it back to pyruvate, and then, using energy, convert that pyruvate back into glucose. This process is called gluconeogenesis, making new glucose.
So the liver turns lactate back into glucose.
Exactly. And then the liver releases that newly made glucose back into the blood, where it can travel back to the muscles or other tissues to be used for energy again. It's a beautiful recycling system.
Wow. Anything else happen to lactate?
Oh, yes. Other tissues, especially the heart and even resting skeletal muscle, are very good at taking up lactate from the blood and converting it back to pyruvate. They can then oxidize that pyruvate aerobically in their mitochondria to generate ATP. So lactate isn't just a waste product, it's also a transportable fuel source, especially during and after exercise.
So the heart can use lactate as fuel.
Very effectively, yes.
Yeah.
Different tissues even have slightly different versions or isoenzymes of LDH that are optimized either for converting pyruvate to lactate, like in muscle, or lactate back to pyruvate, like in the heart. It's all finely tuned.
Incredible. Okay, well, glucose is clearly central. You mentioned our diets contain other sugars too. Fructose and galactose. How do they fit into this energy picture?
Right, good question. Let's look at fructose first. It's common in fruits, honey, and of course high fructose corn syrup.
Very common these days.
Indeed. Fructose is mainly processed in the liver. It gets phosphorylated differently than glucose, using an enzyme called fructokinase to become fructose 1-phosphate.
Not fructose 6-phosphate like glucose.
No, fructose 1-phosphate. Then, a specific aldolase enzyme in the liver, aldolase B, cleaves this fructose 1-phosphate into two smaller molecules, DHAP and glyceraldehyde.
Which sound familiar.
They should. DHAP is already a glycolysis intermediate, and the glyceraldehyde is quickly phosphorylated to become glyceraldehyde 3-phosphate. So both products feed directly into the middle of the glycolytic pathway.
So fructose basically bypasses the first few steps, including that main PFK1 control point.
Exactly. It enters glycolysis after the major regulation point, which has implications for how rapidly it can be converted, potentially leading to fat synthesis if consumed in large amounts. But ultimately, it follows a similar metabolic fate.
Okay, and this relates to a clinical condition.
Yes, it brings us to Candice S, the 18-year-old mention who strictly avoids fruits and table sugar because they make her very sick. She has hereditary fructose intolerance, or HFI.
HFI, what's wrong?
She has a deficiency in that liver enzyme, aldolase B. So when she ingests fructose, she can make fructose 1-phosphate, but she can't cleave it effectively.
So the fructose 1-phosphate builds up.
Precisely. It accumulates in her liver and kidneys, and this buildup is actually quite toxic. It traps phosphate, depleting cellular ATP and phosphate levels. It also inhibits glycogen breakdown and glucose synthesis.
So she gets low blood sugar.
Yes, severe hypoglycemia after eating fructose, leading to tremors, sweating, lethargy, even seizures. It can also cause liver damage, jaundice, vomiting, and kidney problems, especially in infants if it's not diagnosed. That's why strict dietary avoidance of fructose and sucrose is the treatment.
Wow, that sounds much worse than just not liking fruit. Is there another fructose condition?
There is, called essential fructosuria. That's a deficiency in fructokinase, the first enzyme. In this case, fructose just isn't phosphorylated efficiently in the liver. It mostly gets excreted in the urine. It's a benign condition because that toxic fructose 1-phosphate doesn't accumulate.
Okay, so the specific enzyme defect makes all the difference. You also mentioned glucose can sometimes be converted to fructose.
Yes, via the polyol pathway. Glucose is reduced to sorbitol, which is then oxidized to fructose. This happens in certain tissues, like seminal vesicles, which use fructose as an energy source for sperm.
Any clinical relevance there?
Yes, particularly concerning sugar alcohols. In the lens of the eye, for example, if blood glucose levels are very high, like in uncontrolled diabetes, the enzyme aldose reductase in the lens converts excess glucose into sorbitol. Sorbitol doesn't easily cross cell membrane, so it accumulates inside the lens cells.
And that causes problems?
It does. Sorbitol is osmotically active, meaning it draws water into the lens cells. This influx of water causes the cells to swell, damages their structure, and eventually leads to the clouding of the lens that we call cataracts. A similar thing can happen with galactose being converted to galactitol.
So high blood sugar can lead to cataracts through this pathway. Okay, what about galactose then, the sugar from milk?
Galactose, primarily from dietary lactose, which is broken down into glucose and galactose, is also mostly handled by the liver. The goal is to convert it into glucose.
How does that happen?
It's first phosphorylated by galactokinase to galactose 1-phosphate. Then, in a key step involving a carrier molecule called UDP-galactose, the galactose 1-phosphate is converted into glucose 1-phosphate by an enzyme called GALT, galactose 1-phosphate urdylate transferase.
GALT.
That glucose 1-phosphate is then easily isomerized to glucose 6-phosphate, which, as we know, is ready to enter glycolysis or be used for other pathways. Infants, because they drink so much milk, have a very high capacity for metabolizing galactose.
Makes sense. And is there a clinical issue here too, like with fructose?
Yes, a very serious one, classical galactosemia. Think of Erin G, the 3-week-old infant presented with vomiting, jaundice, liver enlargement, and maybe early cataracts.
What's the defect?
She has a deficiency in that key GALT enzyme. So like in HFI, when she consumes galactose from milk, she makes galactose-1-phosphate, but she can't convert it further.
So galactose-1-phosphate accumulates.
Yes, and again, it's toxic. It accumulates in the liver, brain, eyes, and kidneys. This causes severe liver dysfunction, jaundice, cirrhosis, kidney problems, brain damage leading to irreversible intellectual disability if untreated, and cataracts this time due to galactitol accumulating in the lens via that same aldose reductase enzyme we mentioned.
That sounds devastating for a newborn.
It is. That's why newborn screening for galactosemia is standard practice in many places. Early diagnosis and completely eliminating galactose and lactose from the diet is critical to prevent the most severe long-term consequences.
Okay, screening is key. It's clear that glycolysis, being so central, needs incredibly tight control. The body can't just let it run wild, right?
Absolutely not. Our bodies have brilliant ways to ensure glycolysis provides ATP exactly when and where needed without wasting precious glucose. It's like a finely tuned engine with multiple checkpoints and feedback loops.
How does the cell know when to speed up or slow down glycolysis?
It's primarily regulated to maintain energy homeostasis, keeping ATP levels stable. The key regulatory enzymes are the ones catalyzing those irreversible steps we talked about. Hexokinase, or glucokinase in the liver, the main control point PFK1, and pyruvate kinase at the end.
And what signals control them?
The cell constantly monitors its energy status. Indicators like the levels of ATP, ADP, and especially AMP are crucial. Interestingly, AMP levels are a much more sensitive indicator of low energy than ATP levels. A small drop in ATP causes a much larger percentage increase in AMP.
So AMP acts like a loud, low-fuel warning light.
Exactly. High AMP strongly signals the need for more energy production, activating glycolysis. Conversely, high ATP signals that energy levels are good, and it tends to inhibit glycolysis.
Okay, so how does that apply to the specific enzymes? Hexokinase first.
Most hexokinases, the ones in most tissues, are inhibited by their own product, glucose-6-phosphate. If glucose-6-phosphate starts to build up because downstream pathways are slow, hexokinase gets inhibited and the cell stops taking up more glucose. Simple feedback inhibition.
Makes sense. Don't bring more in if you can't process what you have.
Right. But remember the liver's version, glucokinase, it's different. It's not strongly inhibited by glucose-6-phosphate. This allows the liver to take up large amounts of glucose after a meal, even when its own energy levels are high, so it can convert that excess glucose into glycogen or fat for storage.
Clever design for the liver's role. What about PFK1, the main control point?
PFK1 is subject to complex allosteric regulation, meaning molecules bind to it at sites other than the active site to turn it on or off. As we said, high ATP inhibits it. Makes sense if you have plenty of energy, slow down the production line. Citrate, an intermediate from the TCA cycle in the mitochondria, also inhibits PFK1.
Why citrate?
High citrate levels signal that the aerobic pathway is well supplied with fuel, so you don't need to push more glucose through glycolysis just then.
Okay, so ATP and citrate are stop signals. What are the go signals?
High AMP is a powerful activator, the low fuel light we talked about.
Right.
And there's another really potent activator called fructose 2-gummyl-6-bisphosphate. It's not a glycolysis intermediate itself, but its levels rise when glucose is abundant, and it acts like an accelerator pedal for PFK1, strongly stimulating glycolysis, especially in the liver. Its levels are controlled by another enzyme, PFK2.
Fructose 2,6-bisphosphate. Okay, a special activator.
Very important one. This connects back to Otto S. During his sprints, AMP levels rise sharply in his muscles, strongly activating PFK1 to ramp up glycolysis and get that quick ATP.
Got it. And the last enzyme, pyruvate kinase.
Pyruvate kinase, the final step producing ATP, is also regulated. Again, high ATP inhibits it. Don't make the final product if you already have enough energy.
Makes sense.
It's also activated by an earlier intermediate, fructose 126-bisphosphate, the product of PFK1. This is called feed-forward activation.
Feed-forward.
Yeah, it ensures that once glycolysis commits past PFK1, the later steps are ready to handle the increased flow of intermediates. It keeps the whole pathway coordinated. In the liver, pyruvate kinase activity is also regulated by hormones via phosphorylation, slowing glycolysis during fasting when the liver needs to make glucose, not break it down.
Wow, layers upon layers of control. It's really intricate.
It has to be, to match energy supply precisely to demand under all sorts of conditions.
So, beyond just burning glucose for immediate ATP, you mentioned glycolysis isn't just a furnace, it's also like a biochemical carpentry shop, providing building blocks.
It truly is. Its role goes way beyond just catabolism or breaking down glucose. It's central to anabolism or building things up to, like what?
Well, glycolysis provides pyruvate, which is a crucial starting point for synthesizing fatty acids, especially in the liver and adipose tissue after a carbohydrate-rich meal.
So carbs can become fat via glycolysis. Absolutely. Glycolysis also provides intermediates that can be diverted to synthesize certain amino acids like serine and alanine, and also the ribose sugar needed for making nucleotides, the building blocks of DNA and RNA.
Amino acids and nucleotides, too. Okay.
And remember, DHAP, one of the 3 carbon molecules made when fructose 1,4,6-bisphosphate is cleaved.
Wow.
DHAP can be easily converted into glycerol 3-phosphate.
And glycerol 3-phosphate is?
It's the backbone molecule needed to synthesize triacylglycerols, which is the main way we store fat.
So glycolysis provides precursors for fat storage, too.
Yes. And one more fascinating side route, especially important in red blood cells.
Yeah.
There's a little detour called the bisphosphoglycerate shunt, an intermediate from the payoff phase. 1,4,3-bisphosphoglycerate can be converted into 2.3-bisphosphoglycerate, or 2.3 BPG.
2.3 BPG. Why is that important?
2,3-BPG binds to hemoglobin and actually decreases hemoglobin's affinity for oxygen. This might sound bad, but it's crucial because it helps hemoglobin release oxygen more effectively to the tissues that need it. Red blood cells maintain high levels of 2,3-BPG specifically for this regulatory purpose.
So a glycolysis side product helps oxygen delivery. Amazing.
It really is. What's truly fascinating here, when you step back, is how seamlessly all these pathways, glycolysis, gluconeogenesis, glycogen metabolism, the TCA cycle, fatty acid synthesis, are interconnected and finely tuned.
It's a complex web.
It is. From the simple sugar you eat like glucose or fructose to the incredibly complex machinery within your cells, every single step seems optimized for life, for energy balance, for adaptation. Understanding these fundamental energy pathways isn't just about memorizing enzyme names or structures.
Yeah.
No, it's about appreciating the incredible biochemical logic, the elegance of the regulation that keeps us going, responding to whether we're resting, sprinting, fasting, or feasting.
Absolutely. You've just taken us on a deep dive into how your body masterfully generates energy from sugars. We've understood that critical difference between aerobic and anaerobic processes, seen the consequences when things go wrong with enzymes or oxygen supply, and glimpsed how vital this pathway is, from literally powering your brain this very second, to building fats, to, yes, even contributing to a cavity.
A wide range of impacts.
Definitely. We really hope this journey through glycolysis has given you some truly insightful aha moments about the intricate chemistry happening inside you constantly. As you go about your day, maybe just take a moment to consider, what invisible metabolic feats are your cells performing right now just to keep you listening, thinking, moving? It's pretty extraordinary.
It truly is.
Thank you so much for joining us on this deep dive. Keep exploring the amazing world of biochemistry.